Introduction to drug discovery and drug development. So what is the aim of drug discovery and development? So the aim is to produce a drug for an unmet medical need to alleviate human suffering with minimal side effects. So the aim of drug discovery and development is to produce a drug for an unmet medical need, okay, meaning a need for which no current treatment, here we mean medical treatment, exists. Okay, with a view to alleviate, meaning to reach comfort or reduce the suffering with minimal side effects. Okay. So by drug we mean a small molecule or a large molecule or DNA itself or DNA. Okay. So what is a small molecule? A small molecule is a chemical entity having a molecular weight less than 1 kilo Dalton. Okay, meaning less than 1000 Daltons. So then what is a large molecule? Large molecule is any entity above 1000. Okay, so this is above 1000 Daltons. Or it can be a therapeutic that is targeted at the DNA. So DNA can be used for replacement, or it can be a drug that interferes with the expression of proteins. Okay, like say siRNA, microRNA, etc. Okay. these large molecules they are also called biopharmaceuticals. They are also called biologics. Okay. So how are large molecules different? So how are large molecules different from small molecules? In the method of manufacture. Well, the small molecules are usually either a modification of a chemical isolated and purified from a natural product or chemically synthesized or has undergone a semi-synthetic process. So what do we mean by a semi-synthetic process? We mean we derive part of the molecule from a natural product and modify, so subsequently modify the structure. Okay. Or it might be a molecule that is an analog of currently existing drug. So if we modify the structure of the currently existing drug and give it maybe some better pharmacokinetic or pharmacodynamic property, then that could also be a source of small molecules. So then what about large molecules then? Well, large molecules, we need a biomaterial, okay? Or a biologic agent, not a biomaterial. We need a living cell. Okay, this living cell may be bacterial, it may be a yeast, or it can be cells derived from organisms. So we can have human cells. Okay, from the hamster cells. Okay, so cells derived from organisms, we call them cell lines. Okay, so they are basically large molecules then are biologically derived molecules. So what could be the examples? The examples can be the most familiar vaccines. They can be therapeutic proteins. Etc. Okay, therapeutic proteins like, say, example would be insulin, growth hormone, etc. Okay. So those are large molecules. So we have asked ourselves the question, how are large molecules different from small molecules in the method of manufacture? Okay, so large molecules, there is also pharmaceutic differences. The pharmaceutical aspects, aspects would be small molecules are usually given per oral. Okay, they are given through the GIT, maybe as a tablet, capsule. And large molecules are usually parenterals. Okay, so why the difference? Because large molecules cannot cross the biological membranes, which are lipid bilayers. Okay, so let us recap. We have talked about the aim of drug discovery and development. So we said, why do we need a drug? We need a drug to alleviate the human suffering due to an unmet medical need with minimal side effects. So, how is it advantageous? How is it advantageous to humans? Well, before the 19th century, people died of diseases like infectious diseases, like, say, cholera, TB, plague. Okay, these diseases, they killed multitude of people. As a result, life expectancy was really, really less. And today we have increased life expectancy. Okay, but that also has got a small problem. Okay, so increased life expectancy, that has also typically produced some problems. What's the problem? Well, they have increased diseases which were formerly associated with aging. Okay, like our neurodegenerative diseases, neurodegenerative diseases like Parkinson's disease, Alzheimer's disease, cancers, okay, degenerative diseases. Okay, which is a challenge today. So drug discovery has had certain unexpected consequences. One of them being, as we have discussed, they have increased the life expectancy. That could be one. The other disadvantage could be it has changed demographics. Okay. So in the West, there is now a predominance of an aging population, which brings its own challenges. Okay, then we have interventions in fertility. So we have lots of drugs, contraceptives, and so the childbearing age has changed from what was in before 19th century. Okay, so drug discovery has also it also has other consequences. Okay, so large molecules currently are the most valuable drugs coming out of the for the pharmaceutical industry. They're most valuable for the industry because they are very high cost. Okay, as far as the patient is concerned, they are very selective. Okay, and they are relatively safe and they have efficacy. Okay, as a result, lots of research is now happening in large molecules. The technology that has enabled the growth, the technology that has enabled the growth of biopharmaceuticals is recombinant DNA technology. Okay, so the tools and techniques of recombinant DNA technology has enabled biopharmaceuticals to come to the forefront compared to a few decades back. So now we ask ourselves the question, who does drug discovery? Drug discovery and development. So there is the pharmaceutical industry, companies. There are, let's begin with academic institutions. There are pharmaceutical companies and there are biotechnology companies. Okay. So, drug discovery and development, it happens in academic institutions. In academic institutions, usually the drug discovery phase takes place. Drug discovery. Okay. And the development usually happens it's usually done by pharmaceutical companies and biotechnology companies. Drug development. So then we have to ask ourselves the question, what is drug discovery? Okay. So all the steps from defining an unmet medical need to finding out a candidate drug. So all the steps which are involved in defining an unmet, from defining an unmet medical need to the eventual finding out of a candidate drug is called drug discovery. Now, drug development refers to all the steps involved in taking a candidate drug to a registered drug. Registered drug. So what is a registered drug? Which is legally approved. Okay. So all the stages involved in taking a candidate drug to it being approved as a registered drug is called drug development. So drug development phase also includes animal studies. Okay, so we say in reaching a drug from the bench, so the story of a drug from bench to bedside. Okay. It passes through two phases. What are the two phases? Drug discovery and drug development. Okay. Okay. So now the question is, who approves the drug? Who approves a drug? So there are regulatory agencies. Okay. In the US, it is the US FDA. Okay. In India, it is the Drugs Controller General of India. Okay. So what is US FDA? United States Food and Drug Administration. Okay, and what is DCGI? Drugs Controller General of India. Okay, what about say European Union? In European Union, they have got EMA. What is EMA? European Medicines Agency. What about in the UK? So the UK has delinked from the EU. So they have got a regulatory authority that is called MHRA. What is MHRA? It is Medicines and Healthcare Products Regulatory Agency. Okay. And other countries have got similar regulatory agencies. Okay, so they lay down regulations for the drugs which are to be dispensed in their own respective jurisdictions. Okay. So let's talk about drug discovery. Some more points. Drug development. Okay. In an industry, it is looked after, drug discovery and development is looked after by the research and development department and the marketing department. Okay, so they are primarily in the industry. Okay, the department that is responsible for drug discovery and development is the research and development. R&D is an acronym for research and development. Okay. Right. So let's look at some semantic changes. Semantic. Semantic changes. So the 12th edition of the Bible of Pharmacology, which is the Goodman and Gilman textbook of therapeutics, the change has been from drug discovery. There has been a semantic change from drug discovery to drug invention. Okay, so this book has suggested that the term drug discovery be replaced by drug invention. So the logic is this. Discovery is something that is already pre-existing and someone discovers it. Whereas invention is something that has not been there before and you are inventing something new. And so recent literature is picking up drug invention to describe what was hitherto drug discovery. Okay, so there has been a semantic change. Okay. So now we are going to look at what are the current new sources of drugs. sources of drug, meaning what are the preclinical strategies employed for inventing new drugs. Okay. So what are the strategies which are employed? Well, the strategies are called target-based screening. Then there is phenotypic screening. And then there is modification. Natural substances. Then there is a follower. Drug strategy. Then there are biologic based approaches. So, target-based screening. Okay, so what is a screen? Screen is an assay. Okay, screen is basically an assay. So we can do target-based screening. What is target-based screening? Target-based screening is a non-cellular approach. Okay, so we look at certain biochemical mechanisms, and based on that mechanisms, you develop an assay. Then you look at different drugs which give a positive or negative result in that assay. And you do that is mechanism based, so it's called target based screening. The second approach is using a phenotypic screen. So what is a phenotypic screen? So this phenotypic screening is the one that has been used for traditional drug discovery. Traditional drug discovery or classical drug discovery approaches. Okay. So this is also called a top-down approach. Okay. So let us look at an example. Example, you make, say, diabetic rats. You take diabetic rats, and one group you call it the controls, and one group you call it the test. And then you give an extract which can be from a natural product. So what is a natural product? What could be the examples? It could be, the source could be plants, it could be animals, it could be bacteria, it could be fungi, or it could be marine organisms. So any life form, you take an extract, and you administer the extract to test subjects, and in control you give the solvent that has been used for the extract, and then you see whether there is a difference between the control and the test, and you find that in the test the sugar level has gone down. Then you say that this extract has got a hypoglycemic effect. So now you have found an extract that is having some activity, and the technique that you use to gain this understanding, it is called Bioassay guided fractionation. Okay, so your aim is to find out the chemical moiety that is actually responsible for the hypoglycemic activity. The way you do that is by using a bioassay guided fractionation. Okay, so you do a bioactivity guided fractionation. Ultimately, you end up with an extract that contains only one component. Okay, that component you purify, you characterize meaning you try to find out what are the functional groups, what is the structure, and then that becomes your lead molecule. Okay. Then you optimize the lead, then you end up with a drug. Okay. When you do biological characterization, you know that it has got a mechanism of action, and you say it increases the secretion of insulin. Okay, so suppose that is the mechanism of action. Okay, so here what did you do? You looked at a disease model. Okay, when you started, you have got no idea about the mechanism of action. So your starting point was the disease model. Okay. Then you found out that the drug has got an activity in that disease model. Okay. Then you go and find out the mechanism of action. So this is called top-down approach. So approaching a screen from a disease to a mechanism. Okay. So that is called top-down approach. Okay. On the other hand, the target-based approach is a bottom-up. Okay. So what is a bottom-up approach? You look at the mechanism of action, and based on the mechanism of action, suppose it has got a receptor. Okay. You look at the receptor structure, and based on the structure, you rationally design a drug, you optimize it, then use it in a biological model or a disease model, and see whether it has got activity. Okay, so you begin with a hypothesis, and then you get you look at the action. Okay. So that is a bottom-up approach. Okay, then what we can do is, the other approach is to modify the natural substances. When we mean natural substance, we mean purified monocomponent. Okay, we look at the target molecule out of an extract. That is what we mean, the final structural compound. And that you can modify, okay, because it may not have favorable pharmacokinetics or pharmacodynamics. So you can alter this structure a little bit, then you get a new drug. The other way is follower drug strategy. What is a follower drug? There is an already existing drug. And the structure is available. Okay. And the pharmacology is published. Okay. Meaning we know what is the adverse effect of the drug, what is the pharmacokinetics, pharmacodynamics, etc. Everything is known. So now what do? You take that structure of the drug, and you also know the disadvantages from the published data, and you try to modify this structure in such a way that it has got an advantage over the innovator drug. Okay, so the first drug in the class, we call it a innovator drug. So you modify the structure in such a way that it has got some advantage over the innovator drug. Then that is a follower drug strategy. So what's the advantage of that? Advantage of using this approach? Because the toxicity, everything is known. And so the company that embarks on a follower drug strategy, they have got greater chances of success with minimum investment. Then there are the biologic-based approaches, okay, where we use a biological source to modify the drug. Okay. So that is how currently drugs enter the therapeutic space. Okay. So which is the best approach? Well, none of them are the best. So the best approach is one which employs one or more of these strategies together. That gives a better result. Okay, so let us summarize what we have learned up till now. We have learned what is the aim of drug discovery. It is to enter the space of an unmet medical need. The ultimate goal is to reduce human suffering with minimal side effects. And these drugs can be small molecules, they can be large molecules, and they can be DNA, okay, or drugs modifying the activity of DNA. How do we distinguish between small molecules and large molecules? By their molecular weight. Okay, so the cutoff we say 1 kilodalton. Okay, so less than 1 kilodalton or 1000 daltons is a small molecule, greater than that is a large molecule. Okay, large molecules have got different names. They're called biopharmaceuticals, they're called biologics. Okay. And right now they are the most important drugs. They have got certain advantages. What's the advantage of a biologic? They are highly selective, okay, and safe and having efficacy. And we have talked about some details about the large molecules. We have talked about what is the difference between drug discovery and drug development. We have talked about who approves a drug. Okay, so different geographical locations, there are different regulatory agencies which look after the drug. And we have talked about which department in the industry is responsible for drug discovery and development. We have talked about the recent semantic change where drug discovery is replaced by a new term called drug invention. Then we have seen what are the currently available, we have looked at currently available drugs, And we have found out that they have been obtained from target-based screening, from phenotypic screening, from modification of the natural substances, by using a follow drug strategy, and by using biologic-based approaches. Okay, and we have said that target-based screening, it's called a bottom-up approach, and the phenotypic screening is called a top-down approach. Okay, and then we have said that the greatest success would be obtained if we use more than one strategy to look at a drug. Okay, so now let's come to a little bit of economics. Economics and time. Okay. So currently, drug discovery, it's about 3.6 billion US dollars. Okay, and it takes about 12 to 15 years for a drug to be introduced into the market, and the patent life is 20 years. Okay, patent life. Okay, at the end of the patent life, what happens? Small molecules become generic. Okay, meaning other companies can start making the drug, and large molecules become biosimilars. Okay, so there are now some legal changes which allows other companies to start making biologics during the patent life itself. Okay, but then that is applicable to the US market. Okay, so this is the economics of drug discovery. So then the question is, why do companies involve themselves in drug discovery and development? Okay, why do they invest 3.6 billion US dollars in this many years? Because there is a very attractive return of investment. Okay. Return of investment. Okay. So currently, maybe if you look at the best-selling biologic, okay, last year the sale was about 19 to 20 billion USD per year. Okay, so there is a very attractive return of investment, and that drives companies towards investing. Okay. So what's the role of academia? Okay. So here, the academia is usually involved in basic science research. Okay, so what is basic science research? You're looking at mechanism of action. You're looking at therapeutic targets. Okay, and then you publish those. You publish the results are published, and the companies which are embarking upon drug discovery will be reading your results, and if you hold the patent in academia, then you become a part of the economic process. So basic science research is usually contributed by academia. Okay, whereas further things which require an investment, so drug discovery is not cheap, as we alluded earlier, 3.6 billion US dollars. So that might not be within the reach of academia. Therefore, they limit themselves to this area, basic science research, which fall within drug discovery. Okay. And then, in an ideal world, the industry takes up the rest of the work. Okay. All right. So with those very small introduction, let us end this first segment of our lecture.